This presentation will focus on the cell-binding protein lactidherin. Lactidherin is also known as milk fat globule EGF factor VIII, and both of these names come from the fact that lactidherin can be found in human milk. Lactidherin is also a very versatile protein that is found in tissues throughout the human body. This protein contains a C1-C2 binding domain, which allows it to bind to the lipid phosphatidylserine. Opposite to this C1-C2 domain is an EGF-like factor that allows lactidherin to interact with a variety of cell types. Phosphatidylserine is not normally found on the surface of healthy cells. This is because flippase ensures that phosphatidylserine remains on the inner leaflet of the cell's plasma membrane. However, when a cell begins apoptosis, scramblase moves phosphatidylserine to the exterior of the cell. Once on the exterior of the cell, phosphatidylserine serves as an eat me signal that attracts macrophages for phagocytosis. Lactidherin is the transducer of this signal. It binds to the phosphatidylserine, and its EGF-like factor attracts macrophages. If this is not successful, there is a risk of autoimmunity, in which antibodies will be developed against the uncleared apoptotic cells. Lactidherin also binds to extracellular vesicles. This is because extracellular vesicles do not contain flippase, and thus express phosphatidylserine on their exterior. Notably, lactidherin likes extracellular vesicles more than cells, because they are smaller and therefore have a more curved membrane surface, which better suits the binding properties of the C1-C2 domain. In order to study these binding properties, a special protocol is used. A mix of liposomes alone is not ideal, because these are all different sizes. Not to mention, in high concentrations, they may form aggregates. Luckily, lipids will spontaneously form a bilayer around glass microbeads. Therefore, glass microbeads of a set size can be used to form a series of lipid bilayers that can then be run through a flow cytometer. These lipid bilayers can be subjected to lactidherin, and the flow cytometer can measure the fluorescence of a tag on the lactidherin to create a binding curve, like the one seen here. Lactidherin possesses other binding properties than just phosphatidylserine, however. Papers in this area usually refer to the C1-C2 domains as discodin. One paper discovered that the C1 domain is capable of binding to collagen, and revealed that this is important because when lactidherin was knocked out, mice became subject to fibrosis in their lungs. In mice and pigs, lactidherin sometimes has different names. It is referred to as SCD1 in mice, and in pigs it is referred to as P47. These names usually pop up in research papers discussing the reproductive implications of lactidherin. Lactidherin knockout has been shown to reduce interactions between egg and sperm and hinder reproduction. In a recent paper, it was found that lactidherin binds to Lewis X trisaccharides on sperm. The C1-C2 domain of lactidherin works well as a fusion protein. Papers have shown that it can be replaced, or the EGF factor can be replaced with nanobodies, which are similar to antibodies but slightly more robust. This allows for lactidherin to localize extracellular vesicles to various cancers, including EGFR expressing cancers. This has also been replaced with a fluorescent tag, which allows for imaging of lactidherin's distribution throughout the body.